Tissue disaggregation is a vital process in cell culture, enabling researchers to obtain single-cell suspensions from solid tissues. Today, we'll walk through the warm trypsinization method, a technique that utilizes the enzyme trypsin to break down the extracellular matrix and cell junctions, releasing individual cells. Begin by sterilizing all instruments and the workspace to avoid contamination. Warm the trypsin EDTA solution and culture medium to 37 degrees Celsius. Collect your tissue sample under sterile conditions and transfer it to a sterile petri dish. Rinse the tissue thoroughly with sterile PBS to remove blood and debris. Then, use a sterile scalpel and forceps to mince the tissue into small pieces, increasing the surface area for trypsin action. Transfer the minced tissue into a sterile tube or flask. Add enough warm trypsin EDTA solution to cover the tissue pieces. Incubate the mixture at 37 degrees Celsius, gently agitating every 5 to 10 minutes. This step usually takes 15 to 30 minutes, depending on the tissue type. Monitor the disaggregation process under a microscope. Once the cells have dissociated, add an equal volume of culture medium containing serum to neutralize the trypsin. Gently pipette the mixture to further disaggregate the cells, then filter the suspension through a sterile cell strainer to remove any undigested tissue fragments. Centrifuge the cell suspension at 300 to 500 xg for 5 to 10 minutes. Discard the supernatant. Resuspend the cell pellet in fresh culture medium or PBS and repeat the centrifugation and washing steps 2 to 3 times to remove residual trypsin. Finally, count the cells using a hemocytometer and assess their viability with a trypan blue exclusion assay. By following these steps, you'll have a single-cell suspension ready for culture or further processing. Remember to optimize trypsinization time, maintain sterility, handle cells gently, and control the temperature throughout the procedure to ensure the best results.